Welcome. And hey, forget everything you think you know about germs. Today, we're diving deep into the world of the planet's tiniest and maybe most powerful organisms. They're the unseen engines that have been running our world for billions of years.
It sounds like something straight out of science fiction, right? But what if I told you that by learning to read the basic instruction manual of a single microbe, we unlocked a superpower that completely revolutionized modern medicine? Well, that is the incredible story we are going to unpack today.
All right, so to really understand this superpower, we've got to pop the hood and see how it all works. I want you to think of a single microbe as a tiny, microscopic chemical factory. And the operating system running that entire factory, that's something called metabolism.
To put it simply, metabolism is, well, everything the microbe does to stay alive. It's this constant buzz of activity, breaking things down for fuel and then using that very same fuel to build what it needs to survive and, of course, multiply. It is the very engine of microbial life.
And this whole process really has two distinct sides. On one side, you've got catabolism. That's the breakdown crew. They are just smashing molecules apart to release all that precious energy. And on the other side, you've got anabolism. That's the construction crew. They take all that energy and use it to build new cell walls, new proteins, the whole shebang. It's this perfect, constant balance of breaking and building.
So, how in the world does this tiny factory keep everything running so smoothly and efficiently? Well, it uses enzymes. You could think of these as the factory's supervisors, or maybe even molecular matchmakers. See, each enzyme has one very specific job. It grabs a specific molecule, that's the substrate, and helps the right chemical reaction happen unbelievably fast. Without them, metabolism would just grind to a halt.
Okay, so we've got our factory, and we've got its supervisors. But you know, for any factory to run, it needs two critical things: the right kind of fuel and the right working conditions. For microbes, getting this right is literally the difference between life and death.
Now, when it comes to fuel, microbes have a couple of main options. If there's oxygen around, they use aerobic respiration. This is the super efficient, high-octane method. It's a complete breakdown of nutrients for the maximum amount of energy, or ATP. That's the energy currency of the cell. But what if there's no oxygen? No problem. They just switch to fermentation. It's way, way less efficient, but hey, it gets the job done in a pinch. Think of it like a backup generator.
And just like us, microbes can be pretty picky about their environment. They need nutrients to build with, of course. They need moisture to make all those reactions happen. They need a just right temperature, which is often, surprise, surprise, around human body temperature. And they need the right pH level. This, by the way, is exactly why things like refrigeration and pickling work to preserve our food. They create conditions that microbes just can't stand.
And oxygen itself is a massive factor. You've got aerobes who absolutely need it, so if you put them in a test tube, they'll hang out right at the top. Then you have anaerobes, for whom oxygen is basically pure poison, so they hide at the very bottom. And then you have the flexible ones in the middle, the facultative anaerobes. They prefer oxygen, sure, but they can switch over to fermentation if they have to survive.
So, we've seen how they survive day to day. But what makes microbes so incredibly adaptable? It's that they don't just survive, they evolve. And a huge part of how they do it is by sharing secrets that are encoded in their DNA.
Okay, first, a real quick definition for you. The genotype is the microbe's entire genetic blueprint, its instruction manual. The phenotype is what you actually see on the outside, the physical traits that happened because of those instructions. Simple enough, right?
Now, the ultimate source of any new trait is mutation. Think of these as tiny, random typos that can happen when DNA is being copied. Most of the time, they don't really do anything. Sometimes, they're harmful. But every once in a while, a mutation gives a microbe a new advantage, a new tool for survival, which it then passes on to all of its offspring.
But here's where it gets really wild. Bacteria don't just sit around waiting for random mutations. They can actively trade genetic information with each other. They can get new genes from a virus that infects them, they can slurp up scraps of naked DNA just floating around in their environment, or, and get this, they can literally build a tiny bridge, called a pilus, to a neighbor and pass along a small circle of DNA, called a plasmid. This is how traits like antibiotic resistance can spread like wildfire through a population.
Okay, so let's pause. We have these tiny factories that can swap and change their own instruction manuals. This is the crucial point. Once scientists understood these natural processes, they had a revolutionary idea. What if,
What if we could give them a new set of instructions ourselves? And that, right there, is genetic engineering. We're not just observing the microbial factory anymore. We're kind of taking on the role of CEO. We're giving these microbes new genetic blueprints to build things for us, amazing things they would never, ever produce on their own.
And the process itself is remarkably elegant. First, you isolate the gene you want. Let's say the human gene for making insulin. Step 2, you use those same kinds of enzymes we talked about earlier to precisely snip that gene out. Third, you paste that human gene into a piece of bacterial DNA, like a plasmid. And finally, you pop that newly engineered DNA back into a bacterium and just let it do what it does best: multiply.
And this is where the magic happens. That single bacterium, and pretty soon all of its billions of descendants, now has a brand new instruction in its manual. It reads that human gene and just starts churning out a human protein as if it was its own. It's gained a new ability. It has become a microscopic factory for medicine.
So what has this given us? Well, the products coming off this microbial assembly line are absolutely life-changing. I mean, before genetic engineering, the insulin needed to treat diabetes had to be harvested from pigs and cows. It was expensive and not a perfect match. Now, thanks to E. coli bacteria, we have a pure, limitless supply. And it's the same story for human growth hormone, for clotting factors that treat hemophilia, interferons, the list goes on and on.
So, let's bring this all home. We went from the basic nuts and bolts of how a single microbe stays alive, all the way to the incredible technology that powers so much of modern medicine.
The key takeaway is really this. We didn't invent some new form of life from scratch. We simply learned the language of the life that was already here. By understanding their metabolism, how they grow, and their genetics, we've managed to turn some of the oldest organisms on Earth into our most powerful and smallest partners.
It is an absolutely incredible story of discovery and ingenuity, isn't it? These unseen engines shaped our planet's past, and now they're healing our present. It really makes you wonder, if this is what we can do today, what problems are we going to solve tomorrow by partnering with our microscopic allies? What will we ask them to build for us next?